I'd like to introduce Paul Fellner here. He is one of the co-founders, president, and CEO of Revision Therapeutics. Welcome, Paul.
Robin, thank you very much. It's a real honor and pleasure for Revision to have the chance to talk to the Stargard's Connected today, and we look forward to talking about the company. Elise, we can go to the next slide. So a little bit about Revision Therapeutics on this slide. We were formed 5 years ago almost to develop products for unmet ophthalmic needs, but by using a particular approach that we appreciated for the potential for speed of development, and that's repurposing. Repurposing is taking an established drug, one that's been approved, and applying it to a new indication. This means that you can rely on the established precedents of safety and manufacturing that are oftentimes uncertain or risk factors in normal drug development, and then take efficacy data, in our case from a valid animal model, to accelerate the development process. This reduces risk and increases speed and capital efficiency. Our lead product is REV-0100, which is a repurposed cyclodextrin. I'll say more about cyclodextrins in the next slides. The primary target for REV-0100 is Stargardt disease. It also has potential in the treatment of dry age-related macular degeneration. And we do have patents on the formulation and method of using the cyclodextrin for treating these retinal diseases. So even though the compound itself is old, there are opportunities to seek protection. In addition, this product would qualify as an orphan drug and has actually been designated as such by the Food and Drug Administration. And FDA has also designated it as a rare pediatric disease drug. The company has some key milestones right now, building on the foundation that we'll talk about today. We need to do some manufacturing of some additional clinical study material to initiate a phase 1/2 study, and our plan is to do this in Australia. We've already laid the groundwork for that. And then relying on the data we've aggregated to date and the data from that study, we'll go forward with an investigational new drug application in the US and other countries, which would allow us to conduct normal clinical studies. We'll say more about our development pathway later. We could have the next slide. REV-0100 has a unique mechanism of action. We'll explain it further in the next slide, but for now, we want to say that the product targets the key factor in pathogenesis of Stargardt disease, and it's believed to be a factor, as I said, in some forms of dry AMD, which is the accumulation of lipid bisretinoid and lipofuscin within the cells of the retina, particularly the retinal pigmented epithelial cells, or RPE cells. It seems to be the case from the studies that have anticipated our work, that accumulation of this lipid material creates a toxic environment, causing cell dysfunction and ultimately death and loss of central vision through atrophy of the macula and central retina. Stargardt disease, as I'm sure this group is well aware, is the most prevalent inherited macular dystrophy. It's particularly insidious because of the more aggressive form of the disease that begins in childhood. And at present, there is no treatment or cure that's been approved. Well, certainly there are some under study. We could have the next slide. So the way this slide describes the way REV-0100 works and the data for why we believe it would be effective in Stargardt disease. On the left-hand side of this slide is a cartoon, which shows a picture of how you might conceptualize an individual cyclodextrin. It's kind of a conical molecule, a cup, you might say, with an interior that is lipophilic. That means that it readily associates with lipids, oils, fats. The exterior of this cup is hydrophilic. That means it's very water soluble. Consequently, what you can do with a cyclodextrin is carry or host a lipid-liking molecule on the interior, but solubilize it in water. This is an important in drug formulation because a lot of drugs are not water soluble. Cyclodextrins help them become water soluble. This cyclodextrin is used in a large number of products, which is why we're repurposing it, because it's already been developed at high quality for pharmaceutical products. But in this case, it's not an adjunct or excipient. This is the active molecule. And it's the active molecule because our collaborators at Cornell University, from whom we've licensed the patents, demonstrated that beta-cyclodextrins have a selectivity for associating with lipid bisretinoid, and they specifically studied the lipid bisretinoid A2E. They could show that each A2E molecule was bound by two of the cyclodextrins, and that binding would remove the lipid bisretinoid from the cell environment into in vitro, in cell culture tests, into the culture medium, and in vivo, just remove it from the retina. This is where we get to the right-hand part of the slide. What is the real reason why we're pursuing this program, the data from this mouse. So there is a mouse model that has been created to study Stargardt disease. In this mouse, the ABCA4 gene is knocked out. ABCA4 is the gene that is mutated in the most common form of Stargardt disease. And then they also knocked out RD8H. Consequently, as with Stargardt patients, this mouse accumulates lipid bisretinoid in the form of lipofuscin more rapidly than normal mice. Now, lipid bisretinoid fluoresces under blue light, so you can detect its presence through fluorescence spectroscopy. On the left-hand side of the panels below the little mouse there are retinal cells that were untreated with the beta-cyclodextrin. On the right-hand side are retinal cells taken from a mouse that was treated. Now, the treatment was in vivo through intravitreal injection, that is, injection into the eye, as with products like Lucentis and Eylea. And the important feature here is that there's a dramatic reduction in fluorescence. The panel on the left is much brighter because there's much more lipid bisretinoid than the panel on the right. And this evidence of reduction in lipid bisretinoid was confirmed through another analytical technique called high-performance liquid chromatography. So in essence, what the Cornell researchers were able to show is that this type of cyclodextrin had a preferential binding activity with lipid bisretinoid and could affect its removal in vivo. Now, let's go to the next slide. So building on this data, there was additional work done. First, the REV-0100, it's a form of what's called beta-cyclodextrin, was compared with other beta-cyclodextrins, and it showed the greatest activity. All of the beta-cyclodextrins did work to remove the lipid bisretinoid, but the REV-0100 cyclodextrin did it most effectively. As I mentioned, this product is a component of drug formulations already. So it's manufactured at the quality level required by health authorities. That's called CGMP, Good Manufacturing Process, Current Good Manufacturing Process. And also, very importantly, there's an extensive systemic data profile in the regulatory filings, which we can reference. So we're able to proceed with the studies by saying this material has already been shown to be safe in humans and animals, that it's readily excreted without modification from the kidneys, that the systemic exposure is safe and certainly the amount of material injected in the eye would be way less than would be delivered ordinarily through other routes of administration, and didn't show any of the toxicity. Now, we also confirmed that intravitreal injection of REV-0100 is safe. We did this two ways. In the mouse experiments, we looked at both the retina after administration of the cyclodextrin and at the ability of the mouse to see this isn't a test for improving vision, because the mouse actually isn't that affected by the accumulation of the lipid bisretinoid, because a mouse doesn't have a macula. But you want to be sure that you actually don't cause damage and lose vision. And the mice that received REV-0100 did not have any impact on their vision, whereas mice that received a different cyclodextrin actually had some loss of vision. In addition, we've confirmed these data through a more systematic study in rabbits that intravitreal administration has no impact on the physiology of the eye. And finally, we did studies to demonstrate that REV-0100 localizes in the retina and the retina pigmented epithelial cells facing the choroid, which is exactly where it needs to be in order to bind and remove the lipid bisretinoid. We could have the next slide. Now, our product does have some significant differences from the products that are in development, and I'm sure you may be aware of some of them. There are oral drugs that are able to either reduce the amount of vitamin A that the eye takes up or impact the visual cycle so that you do not get the formation of this lipid bisretinoid that is the consequence of mutation of the ABCA4 gene. REV-0100 does not, as far as we know, or any of the evidence seen to date, affect the visual cycle directly. It doesn't have a lot of binding activity with the retinoids that are in the visual cycle. Rather, it binds to the derivative lipid bisretinoids. It's not an analog of a retinoid either, like the oral products. So it has a different mechanism of action by binding or chelating and clearing the accumulated lipid bisretinoid instead of slowing the further accumulation of lipid bisretinoid. That said, it could be a good complement to visual cycle modulators because it is able to remove the accumulated toxic material, while these products could be used to prevent further accumulation of toxic levels of material. And the REV-0100 product has very low manufacturing costs. So in a world where cost sensitivity is becoming more and more important, it has the potential to be a very cost-effective drug because it's already manufactured as a more of a commodity. There are other products that are being developed, and for example, news about the Astellas, formerly Iveric product that has been approved in dry AMD geographic atrophy. It's also being evaluated for Stargardt disease. Those are inflammation inhibitors, and basically the inflammation process causes the loss of cells, but it's triggered by the, we believe, accumulation of the toxic lipids. So instead of putting out the fire, REV-0100 prevents the fire by removing the toxic lipids that cause the inflammation in the first place. Now, ultimately, we would love to see, and we think eventually we may see, cell or gene therapies, but REV-0100 has a place in that world as well. First of all, the technical difficulties of developing a gene therapy haven't been overcome. That's because the gene for the ABCA4 protein is huge, and the current vectors that can deliver therapeutic genes aren't adequate to carry this into the cells. It's also, important in a gene therapy that there are cells to receive the gene. So what REV-0100 can do is bridge to the time we have gene therapies by removing the toxic lipids instead of allowing the normal gene to just prevent their buildup through the standard visual cycle process. And in the context of gene therapy, also reduce the level of toxic lipids before the gene may be administered, because it would reduce the overall, we believe, it would reduce the overall stress level of the retinal cells and allow more effective uptake of the therapeutic gene in the cell. Cell therapies are also being developed, but these are going to require, if they're autologous cells, you'd still have to have some intervention to avoid the buildup of lipid bisretinoid. If they're cells from another donor, then that creates other problems with rejection. So again, ultimately, we believe either cell or gene therapy will be able to assist patients with Stargardt disease. But until that time comes, REV-100 could effectively remove the toxic lipid accumulation. And we had a very interesting analogy to the mechanism of action of REV-100 from one of our potential investors. And he said, well, geez, that sounds like the difference between clearing the blockage in a stopped up sink and just turning down the tap so the sink doesn't overflow as quickly as if the tap was on full. And I think that's a good analogy. There's a real mechanistic or impact to taking out the toxic lipids versus merely slowing their further accumulation, especially if the disease has progressed or the subject has already accumulated a high level of the lipid bisretinoid material. Can you have the next slide? So as I mentioned, by repurposing, we're able to have a more efficient development path. That path includes, if we focus on the bottom part, which is where our attention is going right now, manufacturing clinical product, that's CMC, chemicals and manufacturing and controls in the pharmaceutical industry, to do a first-in-human phase 1/2A clinical study. That's the study I discussed that we want to do in Australia, which would provide then the evidence to support an IND in the US and other countries, moving to a potential registration study, depending on what that first study tells us about the activity of the compound in humans. As I said, we have very strong data from the mouse, but we all know mouse and man have some differences sometimes. We're prepared to initiate that based on the data that we have to date, according to our advisors in Australia. And at the same time, we'll be looking at and may want to do non-GLP tox material for a GLP, which is a good laboratory practice toxicology study, just to confirm the data that we already have about safety in the injection directly into the eye. But the reason for redoing this is simply that the regulators have high standards and we want to be prepared to meet those standards. So if they were not to accept the data that we have, this study would be confirmatory. We could go to the next slide. So this slide describes our scientific advisory board, and we have a number of leaders in retinal ophthalmology with a strong focus on Stargardt disease. And that includes Brian Brooks at the National Eye Institute, part of the National Institutes of Health in Maryland in the United States. Elena Dumitrescu, who's an accomplished professor with a large population of especially pediatric patients at the University of Iowa. Peter Issa, who is a specialist in retinal diseases in the University of Oxford. Hendrik Scholl, who was in the United States, is now the head of the ophthalmology group in Basel, Switzerland at the University of Basel. Rupert Strauss, who is at Moorfields Eye Hospital in London, and Janet Sunnis, who is the medical director at the Greater Baltimore Medical Center. These last three, Hendrik Scholl, Rupert Strauss, and Janet Sunnis, were principal investigators in the ProgStar natural history study of Stargardt disease, which has laid a great foundation for how this disease progresses and how you can develop different endpoints to show efficacy of interventions, therapeutic interventions. Next slide, please. Just a word about the revision team. You've heard about me. My co-founder, Valentina Della Casagrande, has many years of research experience in ophthalmology, both in the front and back of the eye, most recently working on other forms of retinopathy. Chaitanya Badwe, also co-founder, earned his PhD from the ophthalmology group at Cornell Weill Medical. And this is how he got associated with this program. Catherine Gregory is an experienced and accomplished business development officer who has experience both in startup companies and large pharma, particularly Shire, where she worked in ophthalmology. Houman Hamadi is an MD, an accomplished drug developer with a number of products to his credit. And then Elise Chin, who you've also been introduced to today. The next slide. So to summarize, REV-0100 is the only drug in development now that eliminates the toxic lipids. We have patent protection for it. It is manufactured and available at high quality with an established safety profile, so it's highly de-risked. Because the material is available, affordable, and has already established safety profile, there's higher efficiency for conducting the studies and getting to launch. And overall, drug repurposing offers the potential for new commercial models in a world where drug price is becoming more and more important. So REV-0100 is faster, less risk, and it addresses a high unmet need in a disease that you all know well. Next slide. And this is where I say thank you very much for your attention. I'm very happy to answer any questions that you have.
Thank you very much for your really thorough and clear, brilliant presentation, Paul. Are there any questions? You can put them in the chat.
I think that there's one question so far, which is, I think it underlines everyone's keenness to have some kind of treatment available. People are asking, when might this be available in Europe?
That's a great question. And it partly depends on the resources that we have, but our goal is to develop the product in parallel in key markets. So we would want to undertake clinical studies for registration in the United States, Europe, and also Australia, because we will have built on the initial studies conducted there, and seek to get approval as quickly as possible in all of these jurisdictions. And there's great incentives to do so. Under European law, for example, you can get an orphan designation, and under European law, the regulatory exclusivity period is at least 10 years. It can increase with the orphan drug designation, pediatric designation. And so there's a lot of incentive to develop in Europe and in parallel with the United States. So that timeline that I showed, it might be delayed, 6 months or a year, or it could be on the same track as the US.
Thank you so much for that. That's really exciting. So at the moment, there are no other questions on the chat. Does anyone have a question they want to raise?
Yes. Can I ask, how often would you need an injection with this?
Yeah, so what we're anticipating is that we will have an initial treatment phase involving monthly injections. But we expect that once we've achieved a level of lipids that is more in the normal range. And I think it's important to remember that lipid bisretinoids are present for people with all kinds of vision. It's just that they accumulate more quickly in Stargardt disease patients because of the mutation of ABCA4. So there's a level of lipid bisretinoid which is subtoxic. Once we've achieved that level, we believe that we could go to a less frequent dosing regimen, because you only need to remove as much as was accumulated over the past period of time. In the initial phase, that's going to be many years of accumulation, but after that, less. So we expect that we'll have a different profile. That said, it'll be something we'll have to establish through clinical work, and may not be something we know until years after the hopeful launch, through continuing studies of subjects under treatment who get treated less frequently.
Would this carry on for many years for a patient?
Yeah, our expectation is that because we're not addressing the underlying genetic defect, that subjects will have a lifetime of treatment ahead of them. Now, here's where the combination with the oral products may be super beneficial, because you could do the more intensive REV-0100 therapy to get the lipid bisretinoid levels back to normal, and then potentially use the oral products to maintain that lower level, and further stretch out the need for additional REV-0100 treatments. But neither the oral products nor REV-0100 are going to be curative. That will depend, in all likelihood, on development of an effective cell or gene therapy.
So this would be ideal for someone who is newly diagnosed with Stargardt's.
We think it would be appropriate for really any patient who has the potential to preserve vision by removing the lipid material. So yes, somebody newly diagnosed would be ideal, because it's unlikely to have much effect on recovering vision. Now, I would say that there's a potential there, because if the toxic lipid buildup is only causing cell dysfunction, but the cell is still living, removing the toxic lipids may return that cell to normal functional levels. So that could return some normal vision to people with Stargardt's disease. And yes, I think with all of the interventions, you would want to start as early as practical in order to avoid the accumulation of toxic material leading to loss of vision.
You probably don't know the answer to this one, but since this sounds like it's a fairly common thing already, presumably the cost would be reasonably low. I'm just thinking of the cost of hospitals in the UK, the NHS, etc.
So yes, and that's something that we're really concerned with, and using repurposing as an opportunity to address. The actual cost of manufacturing will be low. There will be the usual tens of millions of dollars of cost of development because of the need to conduct the clinical studies. Of course, because the clinical study requirements for rare diseases are proportional to the patient population size. And given our safety profile, we think the risk benefit is going to be likely strong here. There's an efficient path to doing those studies and getting registration. The bottom line is we've modeled this out where the annual cost could be in the single-digit thousands or $10,000 per patient, which would still return a very substantial profit to the company, while representing a especially low price for a rare disease drug product, a new one. And we would be looking at other ways to be even more aggressive on that pricing strategy.
Thank you.
Can I ask another question that's being raised? Is people are very keen to get involved, but they are in Northern Europe. Is there any potential at all for them to get involved at this stage in any trial, any work that you're doing from Northern Europe?
So we haven't fully explored the opportunities to conduct our first human study in Northern Europe. We focused on Australia for three reasons. One, they're very good in ophthalmology. Two, the cost basis is relatively affordable, especially compared to the United States. And three, Australia has a strong incentive to conduct early phase studies there because they rebate about 40% of the amount spent back to the company through a tax credit. Once we've completed the Australian study, which would be, we estimate about a year to a year and a half after we start it, then we'll look at doing worldwide clinical studies for possible registration or adapting a phase 2B into a registrational study. In that case, we would be looking to build out sites in the UK and in Europe. But we're looking, that's still 18 to 24 to 30 months down the road.
Thank you for that. I think lots of people are interested in being involved in any trials that you're undertaking. So the fact that we're in touch with you and we'll hear what you're doing as you go along, we can alert people early to when there might be opportunities for them to become involved.
Yeah, we're similarly really happy to be connected with Stargard's Connected, so that we can get that communication to patients and really so that we can also have direct contact with people who are affected by Stargardt disease, both the patients and their families, and be on track to provide a therapeutic that addresses their needs.
Can I ask a question?
Yes.
Okay, hello. My question is a challenge that normally we face in this inherited retinal disease context, in the rare diseases. When we get something that actually works, we need to get approval for the regulatory agencies in each country or in each region, and then finally try to get in public health and et cetera, et cetera. So in terms of outcome measurements, it's common for us to see that some clinical trials, they actually are working, but the way that we need to prove that they are working for the regulatory agencies, or even the outcomes that we decide, or when the clinical trial is designed, sometimes they don't match. So we have something that actually works for the patients and patients are good with them, but then when you go for register or regulatory agencies, we see a challenge there. So do you think this will be a challenge for the clinical trials you want to start, to go on and to study, or are you, or how are you addressing that to avoid this future challenge?
That is an issue that everyone in drug development faces, which is the need to translate something that works into something that can get approved. And the regulators have very high standards for quality of data and what data they're going to accept to demonstrate the benefit profile that would justify approving the drug. We're in early stages now, so our preclinical data give us a strong indication that we can safely remove the lipid bisretinoid. What we don't know yet is how that will impact human subjects, which is the purpose of the phase 1/2 study that will be conducted in subjects who have Stargardt disease, and we'll be using various measures to analyze benefit that may be resulting from administration of REV-0100. Those will be exploratory endpoints. The main endpoint will be safety and tolerability, but those exploratory endpoints will start to tell us about how we approach the regulatory authorities and put together a plan that provides the data that the authorities need to approve the product as quickly as possible. So we're still a little too early to be able to really address your question because we don't have the first human data to build on to understand how we can relate the benefits to approvable endpoints for the regulators.
Thank you very much.
Yeah, you're welcome. Thank you for the question.
I have no other questions in the chat section. Does anyone else here, any participant want to raise their hands to ask a question? Any participant? Okay, I think there are no more questions. Oh, there's no more questions, but there's huge thanks for all the work that you're doing and a lot of excitement. And please be assured that there are a lot of people who would like to become involved in any way they can possibly. So I'm very grateful that you'll stay in touch with Stargardt's Connected, and we will keep everyone informed through our website and through our regular newsletters.
Listen, we really appreciate the opportunity to talk to you all. We know that you're the folks who are, affected most by this. And if there's something we can do to change the trajectory, we absolutely want to do it. So we appreciate the support. We look forward to staying connected, and wish you all a good day. Lisa and I will be dropping off with the break. But I'm going to read the chat first, because there seem to be some good comments there, and I've been trying to pay attention to the Zoom, so I haven't read the chat.